Yes.
Okay. Because I see that the P53 has a very important functions in cancer disease. So for the DAS3 sigma have a very important functions in regulations of P53. And my question is the first one. Can you give me the general picture about the interactions? I mean the relationship between P53 and that protein. The first questions. Because I see that yesterday you have a slide about protein DAS3 will stabilize the P53. And then today we have that protein induced genes expressions of P53. And then P53 induced expressions of that protein. And then P53 interact with that protein. And can you give me an overview of that? That's the first question.
Okay.
Okay.
First question, there's a lot of answers. Okay. The question about it, about 1433 sigma and the PBD3 interaction, and how do they mutually regulate each other. You're interested, you could look up our paper. It's all been published. Okay. So I could summarize for you that sigma, in a way, is a target of PBD3. And then 1433 sigma do several things. First thing is to inhibit MDM2 mediated PBD3 nuclear export. Okay? MDM2 has several biological functions causing the nuclear export of PBD3, and then also cause PBD3 ubiquitination and degradation. So 1433 sigma was able to block that process. So that's one thing 1433 sigma do. The second thing sigma do to MDM2 is actually it can facilitate MDM2 self-ubiquitination. Okay? MDM2, as you know, is an E3 ligase, but this E3 ligase, we have to keep them in check as well. They cannot be there all the time. So one way to go is the self-ubiquitination. But 1433 sigma could enhance that process, to enhance the self-ubiquitination of MDM2. The way you can do it is because it helps the MDM2 binding to each other faster. Okay? So that's one thing. The second thing. The third thing is that 1433 sigma not only doing these two things. The third thing you can do is facilitate the oligomerization of PBD3. PBD3 is a transcription factor, but it usually forms a homotetramer, meaning that four monomers bind together for the PBD3 to be activated. So 1433 sigma was able to do that, okay? And then by doing so, actually facilitated PBD3 transcriptional activity, meaning that enhance the PBD3 transcription for 1433 sigma gene itself, and enhance its transcription capability to transcribe other target gene as well, including P21 or GATD45. Okay.
The second question is in interactions between P53 and sigma. So can you show me the detailed interactions? I mean, how can they interact with each other?
Okay. The PBD3, as I mentioned, 1433 sigma is a phosphoserine threonine binding protein. We do the peptide sequence analysis. At the end of the C-terminal, there is a site for 14-3 sigma be able to recognize. And then, so that's how 14-3 sigma combined to PBD3.
And what is the consequence of that interaction?
The consequence of that interaction is, like I say, help the oligomerization process of PBD3, but at the same time, sort of keep the PBD3 away from the MDM2 binding. Okay.
And the last question.
Okay, good.
Okay, because I see that the sigma have a very important functions in regulating of p53. So, are there any research about that sigma? I mean, are there any research try to down-regulate or knock out the sigma to prevent the cancer or treat the cancer?
Very good question. Yeah, you already go to the core of what we are trying to pursue. Indeed, we try to understand whether we could deliver the sigma to those cancer cells who had lost the sigma expression. Okay? So the study still in the stage that's quite preliminary, because at this moment to do the gene delivery is one challenge. Okay? People used to using the virus particle to deliver. For example, I mentioned that we're using the adenovirus to deliver the gene. People using retrovirus to deliver the gene as well. But this type of gene delivery system has some concern. Okay? Adenovirus has some immune response and some other side effects. So these aspects sort of fade away now. Okay? Right now, the new challenge is maybe using the nanoparticle. Okay? Many of you heard about nanotechnology. So the nanoparticle is a way to go. See if we could deliver to the target site of the cancer, okay, a particular type of cancer, to deliver directly to that area, and then be able to see how 14-3 sigma could regulate all of our biological function. We have done another experiment to show that sigma has a very important role in regulating MYC oncogene protein stability. Sigma actually, by doing so, caused the MYC degradation. So that's a good sign for these particular gene delivery, because the MYC could not only taking care of the cancer who has defect in PBD3, also to take care of some of the cancer who have a MYC overexpression. Okay. So that's too promising. As for the 14-3 sigma knockdown or knockout, whether it has been done in cell line system, it was done, and then I didn't have a chance to share with you. We found that the sigma lost the sigma in cancer actually caused the genome integrity problem. Okay? If you compare cancers, let me give you the example. The cancer cell we use here is one of the colon cancer cell line called SCT116. This particular cell line, unlike many other cancer cell lines, have normal chromosome number. Very rare. Usually, you take out cancer cell line, they all have a normal chromosome number, not 46 chromosome number. Sometimes they have 3N, 8N chromosome, or even more. In addition to that, sometimes you see a lot of chromosome breakage. And actually that happens if we delete 14-3 sigma in these colon cancer cell lines, supposedly have 46 chromosomes. After deletion of 14-3 sigma, the chromosome number change, increase the chromosome number. Not only that, there are a lot of chromosome breakage indicating that 14-3 sigma is critical for maintaining the chromosome intact.
So, did you do the experiment that that sigma overexpression?
Sigma overexpression?
Yes.
Yes, we do a lot.
And what happened?
Okay, we do overexpress sigma in certain breast cancer cell line that we know have lost the expression of sigma. Sort of doing the replacement, right? And sigma was able to suppress the tumor growth faster, meaning that it is very effective in suppressing the tumor growth, suppressing the tumor's invasion capability, suppress the tumor's cancer energy metabolism. Okay. This part I have not touched yet. Hopefully in the future lecture I will let you know about how sigma could regulate the cancer energy metabolism. Okay. Talk about the cancer energy metabolism. We come back to a very important characteristic description of the cancer. Anybody know cancer hallmark? Everybody know the term called cancer hallmark. Right? Remember the introduction, I mentioned one of the you could follow me in the Twitter, in the Facebook, Cancer Hallmark, okay? Because recently I organized a journal called Cancer Hallmark, but that's not the only reason, but I think everybody, if you're interested in cancer research, you should know the term Cancer Hallmark. Cancer hallmarks describe the characteristic of a cancer. Sometimes the cancer has the capability to have abnormal cell proliferation, evade apoptosis, have the capability to evade the immunosurveillance, have the capability to do the angiogenesis, and lost the genome integrity, have elevated or dysregulated the cancer energy metabolism. So all these together we call it cancer hallmark. And there's another cancer hallmark called the inflammation process. Cancer also could be manifested due to the deregulation of inflammation process. Okay. So you should pay attention to what is the so-called cancer hallmarks. Okay. And you could look up the review by Dr. Weinberg. Actually, Dr. Weinberg recently wrote a review about the cancer hallmarks. If you want to search for it, go to the Google it, cancer hallmark, you will see the article. It's very important to know. So what we have found, the dysregulation of the 43 sigma loss, or if we replace back, it could come sort of regulate these cancer hallmark in a different way. Yeah. Okay. I see that, yes.
Professor,
yes.
I hear you to say about CDK1. It's also called CDC2, but I don't know really the difference between CDK1 and CDC2. I don't know what condition CDK1 becomes CDC2.
Okay, good. Yeah, you try to know the history, and then like I say, it's very important to know the history of all the genes. Sometimes you heard about a gene called BCL, and you don't know why it's called BCL, or why not we call it DCL, or something like that, okay? And then the CDK2 or CDC2, that's for nomenclature. The nomenclature problem is usually a problem because scientist A, when he make a discovery, he publishes his paper, he will call that gene, CDC2, okay? But then the guy, the next one, the B scientist, become another discovery, and then use the same gene, but he sort of tried to follow the nomenclature original gene identified east, so he called it CDC2. So for your A scientist, called CDK1, and the B scientist called CDC2. And in the end, it become a mega it make everybody confused. So all the scientists, they get together to want to have a universal nomenclature. So they say, okay, from now on we don't call it CDC2 in the human situation. We call it CDK1. Okay? So they, based on the different situation, they have these nomenclature sort of everybody will agree this will be the common use from now on. So you see CDK1, CDK2. Although sometimes you see some of the people pop out, they're still using CDC2. So that's the story behind it. I think for scientists it's very important, like I say, you need to know the history of these genes. And then when you follow it, you will have a much better idea. And it's important for the people, the same group of scientists, when they work together, they want to have a better communication. So they want to have all the nomenclature the same. So that's the history behind it. So CDC2, indeed, in human, is a CDK1. Okay? But in yeast, sometimes they're still using CDC2 all the time. Okay? Because that was original identifying yeast, the genetic system. Okay?
Thank you so much.
Sure. Okay.
So I just have one question that is a little bit outside the box. Although you're alright.
No.
So we've studied a lot about cell signaling pathways, right?
Mm-hmm.
And we know we have a lot of knowledge on the signaling pathways, but actually all the cancer that happens inside of our body, we need an environment. Like there's some interaction between the environment and the genes, all the proteins inside of the tumors as well. So my question is there any use if we just study in vitro studies and a little bit on the animal models with that relating to their human bodies? And if there is any solution, which one would you recommend? For example, creating a niche outside that mimics the human conditions inside of our body, etc.?
Very tough question and very important question. I think what you come up with is a very interesting question is that all the study we have in this cell system, indeed, is that applicable when we do, say, treatment strategy, okay? True, the cell culture we are doing, they are not in the right context inside the body, okay? Because the oxygen condition, even the cell attached to the tissue culture plate condition, they are all different from what these cells actually indeed happen in the inside the body. So that's truly a concern in a way that the in vitro study is that really applicable when you want to do the application of what you have found in the cell in the human body. So in addition to tissue culture system, people advance need to using the mouse system to better address that question. Sometimes mouse still not enough. Okay. So for example, we do the so-called mouse xenograft mouse model. Okay. Xenograft. Anybody heard about xenograft mouse model? Those know about xenograft mouse model, raise your hand. Only you? You're the only one? Two there? Okay. Okay. So the xenograft mouse model is a way we try to put the cancer cell in a much, we call it in vivo situation to see the cancer cell behave. Okay. And usually we put this cancer cell, human cancer cell line, onto the immunocompromised mice, such as new mice, for doing this experiment. So that's the reason we call them xenograft mouse model. Yeah, and because the system still have defect because the mouse do not have immune system. So as I just said earlier, the cancer hallmark, one of the characteristics of cancer is the cancer cell could evade the immune response. Okay? So the mouse, xenograft mouse model, you couldn't totally give explanation or give a good model for us to study cancer. Some of the people started using transgenic mice or knockout mice for this purpose, but there's still limitation because the cancer cells are not human. Okay, the cancer cell, you do a knockout, you do a transgenic, they are still deemed as a mouse tumor, not human tumor. But there's still limitation there. Right now, the newest development is using the human-derived cancer tissue or do the xenograft mouse. But there's still limitation as well because, again, these mice do not have an immune system, except the tumor is much more directly from patient, not from cell line. Okay, a lot of people doing the breast cancer, we're using breast cancer cell line, MCF7 or the MDN-V231 or 361. This cell line already established for a long time, and then it's only specifically coming from one patient. Okay, it's a tissue, so it could not account for all the possibility we found in breast cancer. Okay, so the human-derived xenograft tissue model, human-derived cancer xenograft mouse model is one step closer, but still cannot completely explain what has been happening in our body as a cancer cell. So that's a very critical question. And scientists are trying to find a way to better mimic the condition inside the body. So we will be able to find a better way for doing the treatment. Certainly a challenge, and I hope everybody will, there is space and there's room for you to explore, okay? And then I really encourage you to keep on thinking. That's a very good question. And I think I'm very impressed that you could think about this process because truly that's not only us, it's the physician scientists, for us as a basic researcher, we want to understand what's the better way to mimic the cancer growth condition, and in the end, we have to find a better way for the treatment. Yes. All right, maybe we should start. Okay, we save it for the next time. Okay. So we talk about the cell cycle. Right now, it's very important to tell you about the checkpoint control. We have mentioned quite a few about the mitotic checkpoint. And here is one of the checkpoints I would like to mention to you is the DNA damage checkpoint. Okay? Under the normal situation, sometimes our cells have been receiving quite a lot of challenges as well. For example, you're under the sun, you have UV light. Okay, and you go to see a dentist, you have an X-ray film taken, you see the doctor from my X-ray film. These X-rays are causing the DNA damaging our cell. But our cell have a very good system to do the protection, do the repair. Okay? So here, to show you the first slide to tell you, the checkpoint in cancer is very important. And then, as I mentioned, that, for example, the DNA double strand, because the X-ray, okay, they will have some defect, causing the DNA, the genome integrity. But the cell has a system to repair it, okay? And then if the cell did not do a good job, okay, there's a defective checkpoint, okay? For example, the cancer try to make the cell lost the expression of PBD strain, and they will become cancer. Normal checkpoint. These will allow cell to have cell cycle arrest, and they have enough time to do a DNA repair. So this process is very important. Okay? It happens all the time. Sometimes the barbecue meat you eat bringing certain carcinogen into your body. Your intestinal epithelial cell receive this carcinogen, causing the DNA damage in your epithelial cell. Your epithelial cell will have the capability to repair. Okay? If they could not repair, now you know the cancer cell may form, and then in the end, people will get colon cancer. So watch out the things you are eating. Okay, preserved food, do not eat too much. Okay, canned food. Okay, barbecue. Okay, be aware that what you are eating. Okay, the carcinogen